Hello, my name is Natalia Perez. I'm a professor of plant pathology at the University of Florida Gulf Coast Research and Education Center. And today I'm going to talk about UVC for control of mostly strawberry powder mildew. And I would like to start by saying that this has been a great collaborative effort with researchers from different institutions, in particular the Lightning Research Center at RPI, Cornell University, the Norwegian University of Life Sciences, Saga Robotics in Norway, in addition to University of Florida. So just as a background, strawberry production in Florida is very similar to production in Australia, in particular the region of Queensland in Australia, where we produce strawberries as an annual crop. Our planting season is just the opposite as yours. We plant in September and October, and our season goes all the way to March. So it's an annual crop, and the transplants we use are mostly what we call green top bare root transplants, and those transplants are produced in nurseries far from Florida, mostly nurseries in California, Canada, and North Carolina. Even though we have a certification program that we start with meristem plants, they're clean of any systemic diseases. Those transplants are then propagated in open fields for a few generations, and during that propagation in open field, there's of course a chance they will get contaminated with some fungal or bacterial diseases. And here's just an example of some of the major diseases that we face in Florida, which I believe are some of the same diseases that you are facing in Australia. So to control these diseases in nursery fields and fruit production fields, nursery growers and fruit growers have pretty much the same fungicides available, the same different fungicides group. And at first glance, it might look like there's several groups, but as you can see, there's several different combinations of groups, which is not particularly useful for fungicide resistance management at times. So I'd like to spend some time going over the issue that this has created. And the issue is the emergency of fungicide resistance to multiple fungicides and pathogens. And just as an example here, we have now in Florida for a few years already detected resistance in cholecalciferol naclutatum to the quinone outside inhibitor fungicides or distrobulin fungicides, and those are going to be azoxystrobin or pyroxystrobin. And that resistance is linked to a single point mutation in the cytochrome B gene, and which makes all the isolates that have that mutation resistance to all the fungicides in that group. And this group used to be the main tool for management of anthracnose in Florida, and now it's no longer effective. And to make that a little more complicated, our group has been able to get evidence that the emergency of resistance in cholecalciferol naclutatum in Florida is being linked to transplants that came infected from different nurseries regions with strains that were already resistant to this group of fungicide, which has pretty much made that group useless to control in Florida, because by the time we get the transplants are infected with resistant strains, we can't really use that fungicide group to control the disease. Another example is as in Phytophthora, we have been picking up recently some evidence of resistance in Phytophthora cactorum to mefenoxam or ridomil as FRAC group 44. In this case, the resistance has been limited to a couple of different nurseries, so it's not as widespread as in the case of cholecalciferol naclutatum. But again, ridomil or this fungicide group is one of the most important for control of Phytophthora ground rot in Florida. So if we get transplants that are infected with those resistant strains, this fungicide group will no longer be effective. And then there's botrytis, that we make the analogy as the MRSA of plant pathogens, because if resistance is going to show up, it's show up first in botrytis. So there's a lot of reports of resistance to pretty much all the different fungicide groups out there. And again, our group has been able to link the resistance in botrytis with transplants that are coming in infected with resistant strains from the 3 main different regions that produce and send plants to Florida. So I'm saying all this not to point any fingers to the nurseries, but to bring up the issue that led us to pursue what we've been doing and what is the topic of this talk today, which is the UV. The point is there's too many issues out there in nurseries and fruit production fields, and basically we need more tools. We need more tools to address those diseases at different levels and to break that cycle of using the same products in both systems without even realize some of the issues that we are creating. So we got funding a few years ago from a USDA project, and one of the goals of that project was to develop some non-chemical alternatives among other objectives, but one of the objectives was to develop some non-chemical alternatives that could be used either in nurseries or fruit production fields, on both. So that's what finally brings us to our UV project. So UV is not necessarily a new thing. The lamps producing UV light have been around for over 75 years now. It's being used in hospital settings, water filtration systems, and now more recently, since COVID, there's a lot of small apparatus now to disinfect your cell phones and desktops and so on. The UV wavelength that's most effective is, or has germicidal effective, is in the UVB or UVC range. The UVC range being the most effective, and that's within the 250 to 80 nanometers. So the mode of action of UVC is basically it damages the pathogen DNA. And the tricky thing is that many pathogens, they're smarter than we think. They have evolved systems to repair the DNA damage done by UV, because we have UV light, UVA during the day. So the pathogens have a mechanism to repair the damage caused by UVA. So one of the breakthroughs, I think, that allow us to do what we're doing in open field is being research done by some colleagues in Norway who have found that applications of UV at nighttime are more effective, or at least effective at much lower doses, because that mechanism of DNA repair is not activated at night. That mechanism of repair requires the blue or the UVA light that we have during the day, but we don't have during the dark. So is UV effective against everything? Well, not really. It's mostly effective on organisms, either pests or diseases. They're most exposed on the surface of the plant. And among the diseases, powder mildew is a good target for it. Mostly because it's wholly external to the host. It's pretty much on the surface of the leaves or the fruit. It's particularly problematic in nurseries, in our case, in nurseries in California and Canada. So we found that a lot of the applications of fungicides at nurseries that were actually targeting powder mildew is actually what led to the resistance issues that we have with botrytis, because those organisms are coexisting out there at the same nursery or the same places. The other thing about powder mildew is that it's usually more severe in greenhouses or tunnels or any structures where you're blocking UV. Because of that, it's being already used in some settings in Europe for control of powder mildew, and it's much easier, let's say, to set up lamps in a greenhouse facility where you can have an static structures and on a timer to go on and off for several minutes or hours during the night when there is nobody at the facility. The challenge of our project was to try to build a system for our open field production systems in Florida. So to do that, we needed lamps that we could reach the plants in a short period of time, in a matter of 5 to 10 seconds rather than 5 to 10 minutes. And we needed to be able to get a good coverage of the entire plant, because UV is basically a contact treatment, so it's going to kill whatever gets exposed to it. So you need to be able to get a good coverage of the UV light. So as many plants that you get as close to the plant is what is gonna dictate your dose. So the design of our equipment, it was critical that we add reflectors that would help with that coverage of all the different parts of the plants. So in our case for the strawberry crop, that's why the design looks like as it does. It was basically designed to go over the strawberry bed, in this case the dimension of the strawberry beds in Florida. If we were to do this in Australia, we would have to design an arch, an apparatus that would fit the dimensions of the strawberry beds in Australia. The way we calibrate the UV dose throughout the season is basically by adding UV meters in the field and running the tractor at different speeds. In this case, for example, we are targeting between 200 and 250 joules per square meter, and we had to run our tractor around 1.5 miles per hour or 2.4 kilometers per hour. But the meter is really what tells us if we are on target for those applications. So here's some other examples of a different setting that we built in Florida. In this case was to go over a couple of different beds, 2 beds at a time. Again, we had the arch to go over the strawberry plants, and we have done trials looking at doses going from 85 to 200 joules per square meter. The applications were after sunset, as I said, during the dark, and as I mentioned before, the dose is pretty much dictated by the speed that you're running that tractor. In this case here, when we're targeting around 85 joules per square meter, we are actually running those tractors pretty fast, at about 4.5 kilometers per hour. So here is a video showing how it looks like under that array with the reflectors. The noise you're gonna hear in the back is the generator that's running the lamps. Here I want to show some results of 3 trials that were conducted looking for control of powder mildew. Those trials were run by my PhD student at the time, Rodrigo Onofre, And he looked at a non-treated control fungicide standard UV treatments at 85 and 170 joules per square meter with or without a fungicide treatment. The first season there is not as much disease, but all the UV treatments control powder mildew really effectively. The 2 following seasons we had good powder mildew pressure, as you can see on the untreated control. The UV treatments with or without fungicide were as good as the fungicide standard. It's really on the 3rd season where the UV treatments stand out, because during that season the fungicide standard didn't work as well, and we think that has to do with the fact that the fungicides we were using at that time were starting to see some resistance. But the UV treatments with or without an additional fungicide were highly effective in controlling the disease. In addition to foliar symptoms, the UV is also been shown effective to reduce powder mildew symptoms on the fruit. Here's the results from two seasons. The first one was more conducive, there's more disease. The fungicide treatment was effective, as well as the UV treatments with or without fungicide. But the second season, as I mentioned before, the fungicide treatment didn't work as well, but the UV treatments with or without the fungicides were highly effective. We have also looked at applications once a week or twice a week. And the bottom line to summarize this is that applications at once a week can be as good as fungicide sprays, but it really depends on the cultivar you have and the pressure that you have on your fields. I think as a general recommendation, what we have seen is that for cultivars that are highly susceptible, or for seasons where conditions for disease are very conducive, applications twice a week work better than once a week. But if you have a cultivar that's not as susceptible, or conditions are not as favorable, applications once a week are good enough. So in addition to the units that I showed that we built it to test in Florida, there are now many other units that have been built and are currently being tested in other strawberry production areas, including the 2 here at the top that are being built for use in nurseries. The one here in California that was built for a larger bed configuration, and the one here that's for grapes in New York. And we also been collaborating with Saga Robotics to evaluate autonomous robot that they have built, Torvald, and here is Torvald running in our fields in Florida. It basically does the same thing as what we showed before with a tractor, but without the need for that tractor driving at night. I would like to finish with a few take-home messages, and the first one is that we have shown that UV can serve as an alternative to reduce the use of single-site fungicides for control, in particular of powder mildew, both in nurseries and fruit production fields. And that reduction of fungicide use is not only good economically and for the environment, but also and mainly to save the life of those single-site fungicides and reduce that selection pressure for resistance, not only in powder mildew, for all the other pathogens that are coexisting out there in those open fields. In addition to powder mildew, our collaborators in Florida have now data showing that UV is also effective against true-spotted spider mites, another important pest of strawberries. In particular, the eggs are effective. Their data shows the adults are quite resistant to the UV, but the eggs are quite sensitive. And finally, tractor units or autonomous robots are just as effective. The choice really comes down to what's more practical and economical for you. So with that, I'd like to thank you for listening. Thank you for your time. I apologize, I'm not there in person to take your questions, so I'm putting my email here in case somebody has a question they would like to follow up with me. Again, thank you very much.